Today we have with us Dr. Marc-Andre Sirard. Dr. Sirard graduated from veterinary medicine in 1981 at the University of Montreal. He went into large animal practice before completing his PhD at Laval University in 1986 and spent all his professional life working in IVF. He used a laparoscopic approach to perform IVF in cattle and obtained the first test tube calves in 1985 using a clinically usable approach. During his postdoc in the USA, Dr. Sirard co-developed a method to produce bovine embryos by the hundreds using oocytes recovered from post-mortem cows. He came back to Quebec in 1987 and obtained an industrial chair to work on bovine oocytes and sperm in 1990. He founded the Centre de Research and Biologie de la Reproduction in 1995, which has grown to include more than 100 people today. He obtained a senior Canadian research chair in 2000 on genomics applied to reproduction and has created an international effort to define the normal genomic program in early mammalian embryos, which has become an NSERC strategic network called EmbryoGene in 2008. His work has demonstrated the value of controlled FSH simulation to obtain developmentally competent oocytes in cattle, and his current research activities are focused on the epigenetic mechanism, allowing information transfer from one generation to the next. So thank you for joining us. Dr. Sirard, I'll give the go.
Good.
Yes, you're good.
Good afternoon. Very happy to be here, finally invited to Guelph. I don't know what I've done wrong. I know what I've done wrong, and I'm going to explain it to you pretty clearly. Genetic is like a religion, and I don't have the same faith, so that's why I'm not often invited to genetic meeting, because we are trying to create a new religion called epigenetics. And although we have the same ambition of improving the life of cows, we have different rules a bit. This is just a bit different. So I took the advantage of having to meet with Christine and Flavio and this group and Eduardo today to share with you my perspective, my new belief about epigenetics, how it can maybe change the world for cows at least. So I know you're maybe a skeptical public because you're a geneticist, but I'll try to walk my way in through that and try to make you maybe not believers, but at least interested to the process. So we're going to go very rapidly a few bases of epigenetics. My talk today is going to be focusing the first part on periconceptual periods of parents around conception, fertilization, early embryonic development, culture effect, IVF. There's a very good example with that. And now I have a lot of data on postnatal consequences of those change around conception, especially in Canadian IVF animals. You know all this. I don't have to really go to the details, but it's extra information that the genetic information is not containing. The sequence does not contain all the information. There's added information that is part of the environment effect and the epigenetic effect. Now I can even create two different types of E. One is environment, one is epigenetics, and they're not equivalent. They're a bit different one of the other, but one have a lot of influence on the other one. The mechanic of epigenetics, very summary today, there's like three types of things that happens in our genome. Is the one that is short-term, very rapid, and it's reversible, is histone modification. So I go training today, my genes will respond to training, and tomorrow my muscle will have some pain, and it's epigenetic, applied rapidly to a short-term stress. This has value for short-term response, but not for the long-term. We have the microRNA, and there's people here in Guelph like Jonathan Lamar works on microRNA in bovine. I'll have a small part of my talk about. It's information that is contained between genes or in introns that is transcribed and has an influence on gene expression. So it's not a gene, but it's a factor between genes that influence the expression. So if the microRNA are also potentially affected by SNP, so they may be selected before. But this is kind of a rapid effect, context effect, and it's reversible. What I'm going to focus on is mainly DNA methylation, which is the long-term process. It's like data backupping or storing data. And the good and the bad thing about DNA methylation, it's hardly reversible. So that is creating a pattern. That pattern will follow you for life. Those three mechanisms can work sequentially, but also simultaneously on the same tissue in the same individual. Rapidly, to give an image, because people like this type of, it's like if your DNA sequence is like in a book, and you have sentences with information, and when you methylate, you put a carbon on it, it's like erasing or putting a black mark on it, and it's there. It's in all our cells, but some cells have this black mark on it, and the information is not accessible. So that's the principle of modifying gene expression, is simply covering the information with something that the polymerase cannot read. So in the pre-conceptual period, there's two major time at which DNA methylation is erased. I told you this was kind of permanent, but from generation to generation, when we reconstruct our tissue, we have to erase that. The first period is when we create gonads, germ cells and sperm, and testis and oocyte in the ovary. So those cells are almost completely devoid of methylation. Then we create sperm in blue and oocytes in red. Sperm start to be methylated at puberty when the bull, let's keep with the bovine, the bulls start making sperm. But the oocytes will regain its methylation just before fertilization. So the methylation change is still occurring when we superovulate the animal. This is control of ovarian stimulation, or when the animal is preparing for ovulation. So the preconception, like postpartum period, that's where the oocytes will read the environment and finalize this methylation pattern. So that's important for the rest of the talk. The other timing where methylation is erased is just after fertilization. We don't want a sperm program. The embryo does not need a tail. So we have to remove that information and create instruction for embryos. So the sperm DNA is actively demethylated by enzyme. The female DNA is passively demethylated by cell division. It's a bit different. It's different timing, but at the end of the day, you have a blastocyst here that, contrarily to this image, does not have zero methylation. But in the mouse, it's been shown that about 20% of the methylation coming from the gametes is not erased, is kept. And I'll have a proof of that in a bovine in one of our studies. So two periods of demethylation, one complete during gonads formation, one incomplete during blastocyst formation. When we have those embryos, they come with two epigenomes, the sperm and the oocytes, combined together, erase, and then at the blastocyst stage, then we start having more epigenome being created. Because as you realize, all our cells have the same genome sequence, but our cells will have a different function. So those epigenomes are as numerous as the number of tissue that we have. So when we have a blastocyst, we have two tissue. So we start by two epigenome. But rapidly, as we grow from fertilization to fetuses, the different cells will follow different tracks for ectoderm, endoderm, and we'll start having different programming. So the epigenetic layering will be progressive, and at birth you have like 212 different epigenome in your body for each tissue. But these can be influenced, those epigenetic mark, early in the embryonic life by energy, mainly oxygen, pH, transcription factor, the uterine environments, the fetal environments. We know, especially in human, what women eat during their pregnancies, or access or exposure to disruptor toxin will have an impact on the babies, and later in his life, all his life will be affected. So that's the pattern, and what we're going to try to study is not this genetically programmed methylation, which is pretty fixed. is the one that is not programmed so well, that depends on the early life environment that's going to change. So to talk about what's kind of known about the periconceptual environment, we know, since I was even young students, that during uterine life, the environment of the mother will have an impact on the health of the child. If the mother is obese, if the mother smoke, if the mother have alcohol too much, we know there's going to be effect on the baby. So this is known by epidemiologic evidence. Now we have a lot of molecular evidence in the placenta in particular that can predict obesity in a child based on methylation in the placenta. We also have this phenomenon, in vitro babies, which are a bit different than normal babies. The IVF people don't like for me to talk about IVF babies like that, but they all have a bit differences that should worry the rest of the planet, but we'll see why this is not happening. We have now animal study with bovine IVF. I'm going to cover that in the next hour. And we have more and more epidemiological evidence that, for example, preeclampsia, twinning, uterine restriction, all these effects will have a phenotype later in life that are associated with disease. And the more and more we have the molecular signature that creates that associated with the events that is causing it. In our field, one challenge to study epigenetic is we want to separate the genetic effect from the epigenetic effect. So these two examples will probably be very powerful to your perspective. We know that very young bulls have a decreased fertility, mainly associated with poor embryonic quality. So then we have the same male we can use at different ages and have the same genetics, but then have a different outcome. And we're going to see we have the same thing on prepubertal female that we now use for IVF. The younger female have lower embryo quality, but have also a capacity to have a higher number of oocytes. So then we're going to study this in the same animal, depending on age. So it's the same genetic, just at different age. And then we have another model where we can use BHB level of the mother to see how it impacts the early embryos. So starting with the male, so the design is we selected the same bulls. We have ejaculate from 3 different times. This is just the first few ejaculate. Normally, CMEX does not sell them. They start normally selling them at 12 months, but normally they're approved for really use at 15. So we have the same male, use different sample, and we create a dating system, not a dating system. Yeah, we dated system. We have the same female per bull, so the same cow were superovulated 3 times, and we collected the oocyte 3 times and use from the same male-female combination the male at 3 different ages. So they're all siblings. The only difference that we see is, except for meiosis, they're the fact that the age of the male has changed. We've done the epigenetic analysis of the sperm. Then we make embryo, and we do the transcriptomic and the epigenetic analysis to see what is the impact of the age of the bull on the embryo quality. I told you the age of the bull has a negative impact. When the bull is too young, the embryo are not as good as later on. So if we look at sperm, I don't want to get through this busy slide, but in summary, what we have is in the sperm, the DNA of the sperm have lower methylation when the bulls are younger, and the genes that are less completely silenced are gene associated with spermatotility, calcium signaling, G protein. This is associated with fertility of the sperm cells. Makes sense. So we can see that the methylation pattern of those young animals is kind of associated with an immature testis with maybe lower level of testosterone in these animals. So that could explain maybe the fact that these bulls have lower fertility rate. There's not really, there's a decrease in fertility, but also there's a decrease in embryo quality. That's why we went to look at the embryo from the same bulls, and we found A bit more difference in the embryo coming from the young versus the old one, compared to the spermatozoa. But not surprisingly, about 20% of the changes are maintained. So the change that have been carried by the sperm that were associated with the sperm methylation changes are still present in the embryo a week later. So these changes could have an impact on the phenotype we're looking at, which is poor embryo quality. I won't show you a list of genes because that's not the crowd for that. What I'm trying to do is summarize what are the gene effect in terms of transcriptomic. So the embryo, how the embryo is responding, how the embryo is acting when the father is younger compared to the same father 5 months later. These figure, you're going to see it a few times in my talk, is summarizing the 3-4 main pathway, which are the mTOR, which control metabolism. That's how we decide how long we're going to live, how energy we're going to spend. Protein synthesis, which is the most costly thing we do in a cell. ATP generation in mitochondria, and cell cycle, cell divisions. So these specific targets, mTOR, mitochondria, elongation factor, and p53, which is cell cycle, are a key element for the embryo to go fast or to go slow. And the younger bulls make these pathway slower. Like these embryo are not as functional, and I'll follow back what does it mean in terms of capacity later on. So this is the picture we have with younger bulls. Now, okay, sorry. Now we go to microRNA. I don't want to spend too much time. We also extract the microRNA from those animals, and then microRNA are not made in the testis. They're made in the epididymis, and change if you change the nutrition of the bull. And suddenly you can have an environmental impact, and these cells are sensitive. If the bull is younger, it means that the bull is not grown up yet, doesn't have an adult metabolism, have a teenager metabolism, and the microRNA are probably giving a specific signal for that. We extract the microRNA, and then we remove all the ones that are common to the oocytes, because the oocyte is a pool of microRNA, like 20 to 100 times bigger than the sperm. So what a few molecules of RNA coming from the sperm would change? Nothing. So we only take the ones that are not in the oocyte, that are only coming from the sperm, and then we look in other data sets, which are the targets that are present in the form of messenger RNA in the oocyte. So if those microRNA do something, they would do something on those messenger. And then we look in our data set, the one in 2 cells that are associated with better embryonic development, and then extracted what are the pathway associated with those targets of microRNA. And then we look at the blastocyst, what are the transcriptome reading of those targets. Essentially, if the sperm brings mRNA, can we trace the signature, like like criminal investigation, the cause of what's going to happen at the end? And what we found is mitochondrial dysfunction, elongation factor, mTOR signaling. It's the same thing as we were looking at for the DNA methylation. It seems that nature is using potentially 2 different additive mechanisms to have the same effect on the embryo. And if we pull them back, the microRNA has the same type of targets in the early embryo to change the fate. So the effect of young age seems to be coherent with all the nature's capacity to modulate embryonic survival. Let's move to the female side. Very similar design, the same cow, pre-pubertal, around puberty, after puberty, same bull, again, we matched them. And then we cannot do the DNA and evaluation on oocytes because we need 1,000 cell copy to do DNA methylation. And the oocytes has only 4 M DNA, so it's not, we would need like 1,000 oocytes, and that's kind of difficult from one animal in one time. So So we're going to rely on the post-fertilization effect for that. What I can show you is like here we have the effect of number of oocytes according to age. And maybe you don't know, but younger animals have more follicles, they have more oocytes, but not more embryo. The oocytes have a lower quality when the calf is not puber yet, and that is reflected in the fraction of embryo that becomes embryo, fraction of oocytes that become embryo, which is lower. So that's also the same phenotype, lower embryo quality as we have seen in the male. Now, transcriptome, same thing, boring. But when the students came with 3 sets of data, they come with a list of genes, and then you start looking at, oh, I've seen this one last week. Oh, I've seen this one. Oh my God, it's the same thing, except those 2 TGF-beta1 and hepatic nuclear factor 4, which are kind of a bit more specific to the young female effect. But for us, it's very comforting when you have 3 different studies getting you out of 25,000 genes the same 5 pathways that are highlighted by the effect of age of the parents. So is this just an effect of age? Well, let's try another model here. Here I have cows postpartum, day 45, and they're classified with the BHB level. Higher BHB means the cow is still losing weight because there's still lipolysis in the blood. So these animals are maybe making a lot of milk, doesn't matter, but they have a metabolic status that is not completely recovered. They're still losing weight, even at 45 days. And we compare, we superovulate them, we inseminate with the same bull, we make embryo, we do the transcriptomic analysis, and later in the talk, I'll talk to you about calf. But let's first focus with the embryo coming from the BHB cows. Elongation of inhibition of transcription, EF2, mitochondrial dysfunction, mTOR. Now you're starting to learn the words because it's always the same. So it's the same phenotype we have from the tree model that we have used to explain the fact that postpartum cow have embryo with lower quality, and higher the BHB, the higher is the effect of lower quality. Now we're starting to have an explanation. And in that case, we have no IVF, no culture. It's important for the rest of the talk because this is a confounding factor. So what we're saying there is the blastocyst level at conception will have an impact on the embryo programming for the rest of its life. Rapidly, the culture effect. I was telling you about human IVF. We have about 7 million IVF babies born in the world already. Most of them are younger than 25 years of age because this is new technique. And some have imprinting problem, like large calf syndrome. It's called Biquid Angelman in human. But this is like 1%. This is a worry, but not so crazy. But the average have higher blood pressure, higher glucose. And this is very significant at 90 years of age, and we don't have the adults to see if they become diabetics, but it looks like they're going to be more diabetic from IVF than regular babies. The cause of that? Well, some clinics transfer multiple, that's uterine restriction. That's the cause of this in human population. We know that. But there's also the effect of stimulation, the effect parents are older and fertile, the effect of culture, we're going to concentrate on that. Also, the effect of parents being a bit older, they're a bit more obese on average, chemicals exposure longer, etc. But all these factors, when you create conditions of all these things in the mouse model, for example, separately, you do IVF, the mouse have all these problems. So all those different causes of environment changes have at the end the same phenotype or very similar phenotype, which is metabolic disease or metabolic disturbances. That's the human picture. It's getting more and more on the goggle, and the patients are asking the doctor, I want to have a normal baby. The doctor don't know what to answer to that because they don't understand what's the mechanism behind that. In bovine, we've done a very nice study a few years ago with Germany, where we did transfer embryo back in the uterus at 1 cell, at 4 cells, at 16 cells, or we flush them out, and we culture everything to the blastocyst stage and compare blastocyst to blastocyst, simply to show that the longer the embryo is outside of the mum, in either direction, flush in or transfer back, the number of methylation change, in red it's hyper, in green it's hypomethylation, is in the thousands. So the embryo is sensitive to the environment, very sensitive. And it's in the period I showed you at the beginning where methylation is being erased and replaced. Of course, that's a sweet spot or sensitive spot. The embryo is restructuring the genome, and you put that in a soup with something the embryo doesn't know, and suddenly the embryo adapt or try to adapt and create all those changes. So maybe the high glucose baby is somewhere hidden in this adaptation. So I have a lot of other study in vitro showing all the damage we're doing, but for this talk, I just want to focus that what we see as the in vitro culture effect is very similar to the young parents effect and the metabolic stress mother effect. So all those very different environment conditions put an embryo that's going to be in economy mode with autophagy, less protein synthesis, like the embryo believes there's not enough food outside. The mom was starving or younger, or the bull was younger, they've not grown up yet. The repro system seems to, oh, there's not enough food. No, because mom is still growing. There's not enough food for reproduction because mom is growing. So what we can pull out of that is these conditions create the basis of the embryonic adaptation and the phenotype we have that I'm going to show you with IVF babies in cows, what the phenotype is coming in. So now we're starting to have a mechanistic explanation of what people have been looking at after birth, how it's being caused by different conditions before conception. So conclusion before birth, the age, culture, time, IVF, all this changes. But if it's only at the embryo stage, we don't care, because if there's no impact later on, it's just science observation. There's no consequences, right? I think we now have evidence that there are consequences, and we're going to go through that. I'm going to start with the same design that we have here, looking at the embryo were kind of different. So we made embryo transfer with some of those embryos into very similar recipients. And took blood samples from the heifer at about 10 days of age, looking in the blood DNA methylation, if we could see a signature, a pattern that would say this calf knows that mommy was hungry or not in the blood. I mean, septically, we did that with 5 animals per group. So if I talk to geneticists, it's never going to find something with 10 animals. It's impossible. Well, epigenetic is not genetics. We have 100 times less target. They're much more concentrated. They're variable in level of methylation. So we have much more power to make association. So these are the distribution of the differently methylated regions, DMR, in a genome. And not surprisingly, 50% of the DMR are in genic regions, which is not 50% of the genome. So there's a potential non-random distribution of those methylation. And this is not enough to prove it, but this is a kind of an inch that there's a specific target for this methylation. When we look at the methylation that are close to a gene, so we can assess, because intergenic, there's not much you can say about it, but the methylation that are close to a gene and the number of genes that are methylation spot, and we look at what are the main factor it will impact. The first one is metabolic pathways. And these are calves that were born from mother that were losing weight. It's not approved, but it's kind of a evidence that there's something that the blood knows about, and the blood has different type of red blood. The white blood cells, and now we can normalize for the distribution. But the blood, I don't know if you notice, when you have a disease, you're tired immediately. Immunity takes a lot of energy. So our immune cells are very tuned about the energy we have, and therefore they have a kind of metabolic sensitivity of the environment. And when you're if you're losing weight, you're more susceptible to catch a cold than if you're gaining weight. That's kind of epidemiological observation. But now we start having maybe some clue about how the immune system works with that. I have another set of data that is this is also supported by CMEX, that the epigenetic biomarker in sperm may influence the bull fertility. This is all adults, not young ones, but across bull. And these can be transmitted to offspring and therefore influence daughter fertility. The bull fertility paper is out in March. This one is in press now in epigenetics, so it's coming out. So we work with CMEX with their data sets of bull. We use BFI and took the 100 bulls with the most extreme BFI. We separate them, the 6 bull from each hen of high and low fertility, and look for DNA methylation. Same thing for the female. Was not the same bulls that were selected except one. And we classified the bulls according to their daughter fertility, and we look for methylation analysis on both of them. We didn't expect much because normally these factors are covered a lot by genetic evaluation. This is an important inherent trait in your evaluation. So we were a bit surprised to see that. This is a distribution amongst chromosomes. This is the bull methylation changes across chromosome, and this is the daughter. And the first thing you can notice is there's more dots, more red dots at the end, and they're concentrated on the right side. Here is the X chromosome. Here it's a bit small, but it's the Y. Now the Y and the scaffold are better differences in the genome references, so we're getting a much progress. But maybe not surprised. Maybe we should not be surprised because the bulls has only one X and only one Y. They give only one to their son or daughter. So these are inherited without changes. And who was telling me they're not even considered for genetic evaluation because their complete sequence is not perfectly organized yet in the bovine. But suddenly we find that both in the bull fertility effect and the daughter fertility effect, the most abundant DNA methylation marker associated with the phenotype are on the X chromosome. A chromosome that geneticists kind of overlook because of the technical problem of that. So suddenly maybe it's a good place to look, and maybe it's a nice complement of information for geneticists. Oh yes, the X. Oh yes, the bull have all the same X and all his daughter. Nothing has changed. There was no crossing over. That's the same copy. That's why we can define also. When Eve was born, 200,000 years ago, in North Africa. How do we know that? Because the mitochondria that we have in our body are coming from our mother, and we can follow up the lineage of mitochondria of all the women on Earth and look for the common denominator in North Africa, and we can look for number of mutation per 1,000 years, and it's 200,000 years ago. We all have the same mother because of mitochondria. The Y, though, is coming from North Africa as well, fortunately. But Adam was born 100,000 years later, so maybe they didn't really meet each other like they say in the Bible. But what I'm saying is those chromosomes have a lot of power for driving changes, and maybe that's something that's been overlooked with the genetic analysis because they're much harder to work with. While with epigenetic, we see in them a very good marker for. So the bull could be selected for fertility, but also with epigenetic analysis of their sperm to see if they have their pro-positive or pro-negative methylation marks for the future generation, because they're going to be breeding those X and Y down downstream, and maybe we should better know about those. So we had like almost 500 DMCs and DMR. DMCs are specific cytosins. When we group them together, they become DMR if the cytosins are close enough. They have much more power as well because we collect a bunch of information on the region, and this information has more power to change the phenotype than the individual DMC, which are more like SNPs. 252 for the daughter fertility. Most are on the sex chromosome, and these could be, as I just said, complementary tool for fertility association with adults. Now I'm going to switch to IVF phenotype. This is the only study I know from Pete Henson in Florida 5 years ago, where they look in the same farm with similar genetics in the same farm, the effect of milk production of IVF, conventional or IVF or IVP, call them, sex versus embryo transfer versus AI. And for the IVF, it's not significant as much as for the IVP, but there's a reduction in milk production when the animal are coming from in vitro fertilization. So that was kind of a puzzle, and people working in the field, oh, what does it mean, IVF embryo? I mean, we pay money for making IVF. We pay a premium for those embryo, and oh, maybe they're not as good as we thought we should be. So we said, well, in Canada, we have the largest data sets, and we have the information. We have Bobitech, who's making the most largest number of IVF animal. Let's try to compare those to see what we can extract. And that was done with Roger Q was invited as a speaker here in the spring, I think, Roger. So he's a geneticist like you guys, but we wanted to extract the information using LactaNet data. So we use 2,500,000 animal, 4 million lactation, Holstein, between 212 to 18, where that's a bit of limitation, where we had LPI for both the dam and the bull. So we can correct for genetics, or normalize. So we have 300,000 animal. Out of those, we managed to obtain 732 cows that were produced by IVF and had 1, 2, or 3 lactation already done. So it's a very small part of the population, but the numbers are pretty interesting. And then we said, well, there's maybe a herd effect. So we selected only the herds where there was at least 1 IVF animal and 1 AT animal in the same herd. So we can not normalize management, you know how it works, but at least reduce the impact of management on our observation. And then these numbers are reduced. This one is not reduced because we could collect all the farms where the IVF were made. So the first thing we ask is, what matters? Milk. Do we have higher milk, lower milk? And this is uncorrected for genetics. The correction comes in the next slide. So the uncorrected ECM for these animals, this is first lactation, second, third, cumulative, in vivo, ET, IVF. Going up, going up. Of course, the best genetics is IVF. Second best is ET because you're paying more for those technologies, so normally it's what you expect. So when you don't correct for LPI, the ECM 1,000 of kilos of milk is advantages for the in vitro one. So people say, hey, I'm making money. It's good. I'm getting return on my investment. Then we correct, it's gone. It's still there, especially the cumulative, but it's not significant anymore. So it means that there's a price to pay for IVF. And there's something that it's not really clear here, but first lactation is a bit lower, not significant. This one is going up. And we see that also in human babies. As the animal is getting older, the epigenetic signature or consequences are being diluted progressively. They're still there, because even at the cumulative level, but it's not totally permanent, but it's having an effect. And for each stress, for example, we see that on the first lactation, also on the next generation, F2s. We haven't looked at F2s there. We don't have enough of those. But each stress in Florida, they have effect on milk. It's similar to that, but goes to the F1 and then to the F2 animals. So there's a passing over this information. This one I thought you'd be interested because I don't think maybe you've seen it before, but when we look for that, we didn't find it. This is LPI. This is in vivo, 2012 to 2018. This is the rate of LPI gain, whatever, how you normalize that. And you can see that it's higher in AI because you start with the lower LPI value, but your gain is faster. We're still making progress. That way it means it's starting to plateau, but not clearly yet. ET, it's pretty stable. Again, the gain is decreasing a bit. IVF, we start with much higher LPI, but then the difference in gain is almost half versus the AI. So maybe we're getting to the limit of elite selection. We cannot create as much gain when we start that high versus when we start that high. You can probably explain that better than I. Then we look at the fertility of those animals, because there's a fertility problem in cows. So we use DF. This crowd is aware of that. I don't have to explain. And you're also aware that the rating of DF, it's not percentages, the classification. And 1 point here means quite a lot of cows. And here we have the difference in DF. In vivo is pretty much on the LPI evaluation in terms of predicted fertility. Embryo transfer, we have a decreased fertility with that, but even more with the in vitro. 1.34, it's not percentage, it's quite significant in terms of differences. If we translate that in different variable of DF, what can be found out? We find it takes about 3 to 4 days in average for a cow to get pregnant. So it means 1 cow out of 5 will miss the first insemination, will have one more insemination. That's kind of how you can translate that. So there's an effect of those IVF animals in terms of fertility, and this is pretty significant. Another interesting thing for this crowd, I'm almost done, is gestation length. Maybe you have data. We've tried to find the data of gestation length between 212 and 219. It's decreasing by 1 day. And I'm curious to see what's your explanation. Beef on dairy, climate change, shorter pregnancy when warmer, and then there's no clear effect between the different conception methods. They all showing a kind of very similar decreasing in gestation length in Holstein of high quality, but that's not. The baby may not be an Holstein, but the mother having a gestation length measured is an Holstein. So I'm open for any of your comments about how we explain that. Another thing we looked at is disease in Quebec. We have with Lactanet, Balacta, we have recording of veterinary diagnostic on those farms. There is no bar because this is in percentage. It's not highly significant, but it's really giving us information. The difference is not equally distributed. We seem to have less mastitis, more ovarian cysts. Of course, here we have better genetics, more advanced genetics, but we shouldn't have ovarian cysts and higher level of IVF. But if its metabolism changes as we see in human insulin and glucose, well, that's what happened with BHB. Ovarian cyst. The follicle is not growing as fast because the liver is competing for a lot of stuff, and the follicle is lazy, and that creates some of that, but also create retarded ovulation, decreased embryonic quality, and decreased pregnancy rate. So if I summarize the IVF-related phenotypes, higher milk production in general, but not significant when we correct for LPI. Slightly reduced daughter fertility, slower genetic gain, but higher in absolute merit. What's coming up is we're trying to now dissect the effect of sexing of the IVF. Some IVF animal have been done with sex semen, and the effect, and the other effect on daughter health that are not fertility. We're still working on these data sets. So in conclusion, we think we have a signature in embryo for sure. Calf signature, yes. What does it mean? More and more we have epidemiological evidence to show that it's important. The most important determinant seems to be the energy level environment, either of the parents, of the culture conditions. But you're going to tell me in culture, you're not depriving the embryo by food. No, we give too much, way too much. In bovine, the glucose is toxic to embryos. And we realize that we've been overfeeding our embryo just to be sure they have enough by putting too much energy, not enough fat, not serum. We're trying to avoid serum. So it's mainly carbon. So we're twisting those embryo. And what's driving the epigenetic? Acetyl-CoA that goes for histone acetylation and methylation. They're all controlled by the mitochondria metabolism, which is dependent on how much glucose, pyruvate, lactate you give those embryos. Now it's obvious because we've learned and then we have dissected the process, but now we realize we are spoiling those embryo by giving too much energy, and they feel like they don't have enough because they're totally in the wrong environment. They're trying to adapt, and then they become embryo that are similar to the one with the animal that we're starving. So we're, the most important adaptation is economy mode for almost all the stress we're exposed to. And postnatal phenotypes are real, but we have to keep collecting the data. It's like you guys in genetics, the more data, the more power. So we still have to have more information to, and the project I'm doing here with Christine and Flavio and Elsarp, we're getting really intense phenotyping and epigenetic analysis and UBC, and now I'm going to try to do something with Eduardo to use the one of his herds. He did an experiment to try to provide even more information on how these environments are creating a future mother that will have a predisposition, a trajectory towards higher milk production or higher lifetime versus the other ones. So I want to congratulate all the students who work in epigenetics. A lot of collaboration around the world here with people in CMEX and Bobitech who have provided the semen, embryos, OPU, and access to data. CMEX is not very far from here. That's the only company I know and have accepted to open the black box of epigenetics, knowing that we're publishing, knowing that they're going to show the rest of the world that IVF has an effect. But because we have the information, now we can start acting upstream much faster than the competition. So CMEX has been learning by being open. Maybe there's a good return in terms of capacity to evolve and adapt to this new world, dairy genetic environment. That's it. Question?
Thank you, Marc-Andre. So just correct me if I'm wrong, you did say that higher methylation when the embryo is out for longer, so the period between when they take it out of one animal and put it into a second one, that time period, the longer that is, will allow for higher methylation within it?
No, the longer the embryo spend in vitro, in the dish.
Okay, so the longer it's in the dish, the longer.
Because some were 2 days in the dish, from 3 days in the dish, from 4 days in the dish. They were either flush at certain times or transferred back to the oviduct by surgery at specific time. And we collect all the blastocysts. So we compare blastocysts to blastocysts at the same time. And then we realized the longer they were not in the cow, the more disturbed they are.
Okay. So my question, did you look at any like frozen embryos, or does a freezing process, would that have any effects on anything, do you think? Yeah. Or
there's a bit of an effect because the mitochondria gets highly distributed freezing process, and it takes a few hours for them to restart their machines. And in human, the frozen embryos, if I'm correct, which side? Larger baby or smaller baby? I'm not sure, but either side, huh? I think regular IVF are smaller, and the frozen, I think, are larger. Yeah, I think you're right. But now they have numbers in human, and but now in human, there is so much advantage of what they call freeze-all, because they give drugs to the ovary, and uterus is not insensitive to all those drugs. So if you put the embryo in the same cycle, the uterus was exposed to the increased estradiol and all this, the embryo is kind of still on a hangover. And then they freeze all the embryos. They create another cycle with no stimulation, normal hormonal level, and they transfer back the embryo. So now we have almost all IVF babies are all frozen.
And do you think you're going to see a similar effect, regardless though of the frozen versus fresh in cattle specifically, or do you think that's another study that needs to be done to individually look at that?
I have data that I cannot show and just share with you with CMEX Bovitec, and they have changed over those years from fresh transfer to freezing, embryo freezing, with different change in culture media, but we don't see differences in the output. It's not enough because we have less animal for the second and third lactation in the first half. But these animals, when we compare to similar animals in the second half, like from 2015 and late and more recently, we don't see a freezing effect, if you might say. It doesn't mean it's not there. We don't see it with the number we have.
Perfect. Thank you.
Getting overfed before for insemination or when they are early pregnant? Because there I was asking myself, you want to have the calves gain as much as they can until the 6th month, and then you put them kindly on a diet for them to be fertile still. But could you go more into detail on this half sentence instead?
Yeah, I'm not sure. So what we know is if the animal is prepubertal, quality is lower. So Bovitec were kind of worry about that. So we said, well, maybe it's the nutrition. So we look at follicular fluid, fatty acid, and there's differences in those 8 months because they're babies. And they have adapted in nutrition patterns that create a less differences in energy balance in those younger animals. They're not fat, they're just better followed up. And now they're doing the same for the bulls. You wonder why, eh? And so now they realize that bull nutrition is so important because those factors could be playing against them later on. And now, in my days, bulls were raised on farm and sold to CMEX. Now it doesn't happen anymore. So CMEX raises their own bulls. So now they have control. And this information is important for them to realize it's worth, the extra concentrate value and everything to have the best nutrition scheme. So those animals get to puberty a bit earlier, but also to get a more adult metabolism when they start collecting them. The young animals?
Yeah.
These animals have different level of concentrate already. For they are synchronized with PRID, even if they're not cycling, just to organize the endocrinology pattern. So adjusting the nutrition so they're for 1 month or two, because they collect embryos from, let's say, 6 months now to 8, 60 embryo, 5 flush, select the 10 best they do genetic analysis with biopsies. select the 10 best bulls or female and transfer only those one who have a value suddenly goes up like a few $1,000s immediately just by genetic selection. So what they want is to have a donor that is in the right metabolic pattern, but they're not, it's not really the amount of energy, it's the, say, combination of different type of fat, fatty acid, saturated, non-saturated, that's what we found, that try to create a better stabilized environment for the ovary to believe that the puberty is coming. Oh no, it's not perfect though, we still see differences. But now the rest of the world is copying Bovitec because Bovitec and CMEX used to have like almost a 6 months advance by using technology with younger animals. Put bulls on the list faster than others, but the others say, oh my God, we have to do the same, and then they're doing the same.
Like the chromosome works in a different way, or you can analyze with like the autosomes.
No, it's included in our dragon blasting sequence. It's as an autosome. The Y is different. We have to, in the mitochondria, we also do a methylation analysis of the mitochondria because I'm now a fan of mitochondria. And mitochondria has a genome and it's methylated. And it's coming from the mother. And it contains metabolic information for the next generation. Hidden in this mitochondria, not this small, we have 200,000 mitochondria per oocyte. So that's 200,000 copy of that genome, which is very small, but that's quite a bit of influence. And it's involved. I can tell you that we have now data showing that it's playing a role in metabolic programming. Yep. I mean, it's bioinformatics. They do their magic, and I have to believe something. But the more I learn, the more I realize that bioinformatics, if you don't get involved closely with the bioinformaticians, they do the protocol that they were told, or what is normal stuff, and they don't initially realize the research question you're asked. So the more you interact with the bioinformation, the more you realize that there's a lot of way of doing things, and what depending on the question, then you learn that it should become part of our teaching. You know, for grad students now, they should all be able to do bioinformatics to a point where they understand what the other guy is talking about. Otherwise, it's dangerous. Yes.
I think this could be associated with the decrease in the longevity of the cows as well. Like we have less mature cows nowadays, and younger animals has like a short gestation.
That's been Roger Q has normalized for that, exactly that, because as the animal is aging, the gestation changed a bit, and it was not, it did not correct that. But it was a good idea to see the fact that we're having more an average of 4 years versus 5.5 years a few years back. Yeah, the turnover is crazy now. We have to stop that, and one way to stop it is to breed animal that lives longer and breed less animal for replacement. So there's not a line waiting to push to get a cows outside of the herd because we've paid so much to raise those new cows that we want to use them, but it's not so profitable when we accelerate the turnover process, as Eduardo would say.
This methylation being a bit more permanent process. And now I don't remember if it was on your talk, on your discussion before, you mentioned about 95% of
Yeah.
the methylation kind of fits for all cells, if I remember correctly. And
Yep.
5% a little bit more, I don't know if authenticity will be a good word.
Yeah.
So if a researcher has a question related to, then he believes that genetics is important, but as a
transient mechanism, not a permanent one, is still valuable to investigate those methylation to 5%, or you would recommend to focus on microRNAs, focus on histone acetylation or other mechanism, because it's probably not the case.
I'm totally convinced that the microRNA and histone are dependent on DNA methylation to start with. So that's what is inherited from cells to cells, from skin cells to skin cell, is this epigenetic pattern. And also 20% is transmitted down the generation. What I said this morning is 95% of the blood methylation doesn't seem to vary across animal, only 5%. Because 95% is creating a blood cells with the blood genes we need, and all the other genes like the liver cells or the eye genes, whatever, have to be erased because the blood doesn't need, transparent skin like we have in the cornea. So all those genes has to be locked down, and this locking down is 95% of our genome. The 5%, it could have been more because the white blood cells probably use 20% of the genes that we have, like 7,000 to 8,000 per day. And therefore, it could have been all those genes that are variable. No, it's a subset of those genes that are variable that are maybe sensitive to the environment. The other are genetically programmed during development to be skin or whatever cell. It doesn't change. Erasing methylation takes a specific enzyme that is only active in the oocytes almost, and in the gonads. The other cells don't express the TET gene, which is. And the reason is we don't want the cells to go, haywire. And cancer is part of that. If you lose methylation, or if you gain methylation on P53 in your cancer, you have a metastasis and you're dead. So it's not a mutation. It's only an epigenetic change in the cancer cells that creates malignancy in many cases. It's not a second mutation. But the 5% we have left, we're starting to dissect how many sites out of the 50 millions we have, out of 3 billion, how of those sites are close to SNPs or not, and therefore is there a genetic sensitivity for those sites? Maybe there is, but there's also a portion I think are insensitive of the genetic environment, and they're sensitive to the environment, but not the genetic makeup. I shouldn't say genetic environment because then confusing. So working on those 5% is much easier, much more powerful for statistical analysis, and therefore we can get much more rapid gain of understanding. The epigenome does not change the eye color or whatever. It only impacts short-term natural capacity to adapt, which is mainly metabolism, mainly. Glucose, insulin, mTOR. This is where our system is flexible because it has to be. The rest, we don't want our system to be flexible. And that's where genetic is a bit weak, is fertility, disease in the white blood cells, because these factors depends on the early environments that creates a setup. The cells adapt and prepare. So that's where the irritability is so low for these. And that's why we find some of those factors in our study, because that's where the variation is created by the epigenome. So we should focus on this 5%. Not within a lifetime, but what we're trying to do now, even with an hospital in Montreal, is trying to finalize our bovine model to predict fate at 6 weeks, at sevrage at 2 months, 60 days. And if we're capable of using the blood cell as a key factor to control resistance to disease and on what we don't know, then we have a potential way of predicting fate, explaining the disease later on, and there's a lot of interest on how we can look at that. If it's epigenetic, as you just said, it may be modified. difficulty. So for example, if you take an IVF baby, I'm going to use this example. Take an IVF baby and you put one baby on a formula, the other one on breastfeeding. There's an effect. You can reverse the high glucose effect, not totally, but partially with the breastfeeding compared to formula. So the nutrition can have controlling or dilution effect on the epigenetic changes. So that's what is important for human and animal. If we can act early, like in humans, if you have a genetic disease and we can tell you're going to take a blood sample and we're going to tell what you're going to have as disease, you don't want to know. You don't want to know how often you're going to die because there's nothing you can do about it. But if we can tell a baby at birth, well, your baby should not have formula, should not have fructose in his juice, should not, because he's already prone for higher glucose, suddenly you can do something about it. And the best model is the bovine, because we can have defined environment, we can change the environment, change the methylation mark, change the fate. We can create a system where we're gonna on purpose try to do that, changing the fate, for example, for those 60 days. We shouldn't keep that animal unless we change the feed of that animal to reprogram, or we give the animal vaccination or whatever, because it's the immune system that is weak. So there's a lot of potential if you know how it works.
Nowadays to change the methylation in young or adult animals, or should we make some intervention during the embryo stage?
Yeah, we're working on intervention on the embryo in vitro by changing the media, the methyl donor, things like that, because we know we have the system. I'm doing that with Brazil, so they're very advanced in metabolic analysis, and that's a way. We can use CRISPR with not a replacing enzyme, but a demethylation enzyme, and we can target a site. Now they're doing that in stem cells for specific disease analysis, so it's doable with CRISPR, but I don't see, a cheap way to do CRISPR modified methylation in animals. We can use food. I mean, it's food and selection. Food to try to change what's changeable, but selection to remove the animal that are already in the wrong track. They have no colostrum for whatever reason. They have this, they have that, but they look okay at 60 days, so you don't know which one to keep. Suddenly you do the blood test. Oh my God, this one is doomed. It won't go anywhere, and we have a risk factor sufficiently powered to make the call, and then to have less Hereford to be raised, but the right one that's gonna live one more year. That's the goal. One more year of life. If we can do that, we win. Well, thanks for the discussion.